Hello, everyone. Welcome to our webinar series, Basics of Next Generation Sequencing and its Application in Precision Oncology. We're about to dive into the third and final webinar of this series, titled Bioinformatics Analysis and Reporting for Comprehensive Genomic Panels in Precision Oncology. I'm Tony from Roche Diagnostics, where I am the Scientific Affairs Partner for Sequencing. I'm thrilled to be your moderator for this webinar, which is presented by LabRoots and proudly brought to you by Roche Diagnostics Medical and Scientific Affairs. We highly encourage you to participate and stay engaged by submitting your questions throughout the presentation. Without further ado, let's give a warm welcome to our esteemed speaker for the series, Dr. Jeremy Segal, Professor of Pathology at the University of Chicago, co-founder and president of the Gold Consortium. Dr. Segal, I'll hand it over to you.
All right, hi there. Thanks for the introduction, Tony, and welcome back for the third and final part of this series on next-gen sequencing and personalized oncology. These are my disclosures. They're the same as we had last time, and so I'm just going to get started. Last time, we talked about all of the wet lab considerations related to oncology NGS panels, and this time we're going to cover the downstream aspects, which include the bioinformatics, the computational options, and the reporting. And it's really a lot to cover in an hour, so let's dive in. To recap quickly, we ended the last lecture with the sequencing itself. The sequencer, it just takes in a large number of molecules, and it gives us sequences of a representative subset of those molecules, and we can tailor it to get as many reads as we want for a particular assay, for a particular patient, by carefully selecting which flow cell we use and proportionally loading the samples in order to get the number of reads we want. But the sequencer, it doesn't just give us the sequences only. For every base that it calls, it also estimates a chance of error, depending on how good the data looks. And that's called a quality score. And it's usually a number between 0 and 40, with 0 being horrible and 40 representing approximately 1 in 10,000 chance of error. And then these numbers are each encoded into a single character. Our final common sequencing output that we can start working with informatically is called the FASTQ file. And it's a repeating set of 4 lines. We have a header line, which contains information about the sequencer and the flow cell, and also, most importantly, includes a unique X and Y coordinate from the actual flow cell that delineates this particular fragment of DNA. And then we have the sequence itself, and we have this plus sign, and then at the end we have the quality scores recorded for each base call. And then, obviously we have a ton of these for all the molecules that we've sequenced, and that's our FASTQ file. So now let's imagine putting together a little pipeline to process this data with different steps, and at each step we're going to get a different file output. And we'll focus this on variant calling. So we may have multiple steps, including some upfront steps to do things like removing adapter sequences. But there's really 2 key steps in this process. The first is aligning our data to the genome so that we can order and review it. And the next critical step is looking at that aligned data to call variants at different genomic positions. So we'll start first with the alignment step. There are many algorithms out there to align data to the reference genome. Ultimately, what you want is something that's fast, and it shouldn't be a big compute hog. But by far the most important thing you want are good quality alignments. Different aligners may be better or worse for certain types of assays. For example, we use NovoAlign for our small amplicon panel because it has nice features for automatically removing primer sequences from your data. But for the hybrid capture panels, far and away the most frequently used algorithm in labs like mine is the Burroughs-Wheeler aligner, or BWA. And specifically a newer version of that aligner called BWA-MEM, where MEM stands for Maximal Exact Match. A discussion on how these aligner algorithms actually work is kind of beyond the scope of this lecture, and honestly, I'm not the best person to ask about it, but I can show you how you use one. If you don't know how to use a piece of software, you can do what professional programmers do, and you can Google it. So we can look for BWA-MEM instructions and get to the instruction manual. And if we look it up, it'll actually give us this particular line of code to use to align our data. And the simplest way to use this software is to be working in a command line Linux system This is how most bioinformaticians work most of the time. It's just a text-based operating system. So it's just a text-based way to use a computer. And the basic things to know are that you're always present inside a folder, you're in a location, and you can move around up or down or between folders, and then you can run instructions. So for example, you can type ls to see what's in the folder where you're at. And here we can see in this folder where we are, we have the HG19 reference genome sequence, and it's conveniently already been indexed to work with BWA, and that's what all these other files are. And we have our read1 and our read2 FASTQ files. Remember, with the large-scale oncology panels, we're typically dealing with paired end sequencing data. That means that for every molecule that we sequence, we're getting 2 sequencing reads, 1 from each end. Here's our patient DNA insert fragment with adapters on either end. And what we're going to do is we're going to get a sequencing read from the left side and another one from the right side. And we're going to put them into 2 separate files, a read1 and a read2 FASTQ file. That's why both of these files are right here. And if we have these files here and the reference genome, and we have BWA installed on our computer, we can just type BWA mem and then the location of the reference genome file, and then the locations of the 2 FASTQ files, and then this little caret symbol, which means we're going to output the results to a new file, and we'll call that sample.sam. Then we press return, and it runs. And now if we type ls again to see what's in our folder, we can see that there's a new file here. That's our sample.sam that we just made, and it's going to have our aligned data in it. So let's take a look at that file. Here we can see the first sequencing read of each of our 2 FASTQ files, read1 and read2. And in the SAM file that we just produced with our aligner, they're still here as the first and the second aligned sequences in the SAM file. The SAM file is just an interleaved file with matching pairs of reads from the FASTQs merged into a single file in the same way as if you were like shuffling a deck of cards. So now we can see here that we still have the unique identifiers from each of the FASTQ entries, including the unique X and Y coordinates for that molecule. But we have a number of new features here as well that the aligner produced. So first and foremost, we now have a mapping location within the genome where that sequence has its best match. And notice that these 2 numbers for these 2 reads are really close to each other. And that's what we generally expect. So if you just happen to take a fragment of DNA from a sample and you look at the 2 ends of it, unless something really weird is going on, they should really map at positions very close to each other in the genome. And if they don't, it would be unusual. Here we have mapping confidence scores. A top score is 60, indicating the aligner is very happy about having only 1 unique place in the genome where this read can go. And a bottom score would be 0 or a total lack of confidence that the read has been assigned to a unique location. If you're dealing with multiple copies of a gene or pseudogenes or repetitive DNA elements, then you'll end up with a lousy score. And lastly, we have these alignment information strings that tell us details of the actual alignment of our reads against the reference genome. In this case, our reads are 80 base pairs long, and it says 80M and 80. And that indicates that the read sequence is a perfect match against the reference genome for all 80 bases. So if we compress our SAM file into what's called a BAM file, and that's all that the BAM file is, it's just a compressed SAM file. It contains exactly the same information. Then we can load that BAM file into a viewer, like the Integrated Genomics Viewer or IGV, and we can actually look at our sequences. So here you can see the read1 and read2, and we can see that they both map to a region right around exon 2 of the KRAS gene, which is down here. And because they're all gray, we can see that they match with the reference genome here perfectly, and that's what they should, because they were perfect matching sequences. Okay, so that's that. Now let's do something different. Let's add a mutation into read1 here. And we'll throw these 2 new reads back through the aligner and see what happened to the alignment strings. The first one here is unchanged. It still says 80M. And that's a little counterintuitive because there's a mismatch, but this first string happens to be only about whether there's an insertion or deletion in the sequence. The second one, though, called MDZ, it does highlight a sequence difference. And what this is saying is that there are 45 bases that match perfectly with the reference genome at this position. And then we're supposed to have a C, but we don't. And then there are 34 more matching bases. If we look at this alignment in IGV, we can now see our C to A mutation that's present in the viewer, because the viewer is taking advantage of the sequence information and the alignment information that's present in the BAM file. Now, let's put in a 5 base pair deletion into one of these reads and see what happens. After the alignment now we do get a disturbance of this first alignment string. This is saying that there are 34 bases that match the genome, and then there are 5 bases deleted, and then there's another 46 base pairs that match. We get a similar, more specific result in the MDZ string there. Notice our mapping quality score also went down. The aligners are really unhappy with adding indels, insertions and deletions into their alignments, and they penalize themselves, which can be a problem. But now, when we load this BAM into IGV, we can see our 5 base pair deletion here in the alignment. So now we'll take these concepts and we'll put them to work for variant calling. So here you can see an IGV window, and we've loaded in all of our aligned hybrid capture data into it. And now we're just going to do variant calling. So this is a stepwise procedure where we're going to go base by base by base by base, one at a time. And for each base position, we're going to add up all the variant signal that we see by looking at one read at a time and tabulating the results. So for each read, the software identifies it as containing pertinent data for our position because we have its mapping position, and then we can look at the alignment to figure out the base call at the exact position that we're interrogating. So in this case, the base call at that position is a wild type cytosine. So we're going to tabulate, because it's a perfect match, and we're going to tabulate a C at the bottom and the corresponding quality score like this. Next read, same thing. We have a wild type base, so we'll add that. And then this next read, we actually have a mutant base call at this position. So we'll tabulate a T with its quality score, and so on and so on. And what we've made here at the bottom is called a pileup. And it's an important part of most variant calling algorithms. And at the end, a variant call decision will be made for each genomic position in our assay using this pileup information. And it may or may not include a p-value based on the level of evidence, et cetera. I do think it's generally a good idea to have some kind of system that upfront eliminates bad quality base calls and works with only the good stuff. As a clinical lab, we should be focused on producing a reliably satisfactory amount of data for each sample. So we should be in a position to comfortably let the low quality stuff go. And I think that's the best way to get high quality results, and it's going to make both the validation process and the clinical service run more smoothly. There are many algorithms to perform this type of variant calling procedure with, and people debate a lot about which algorithms are better. But honestly, I think they're not all that much different from each other, and that you can really make an assay work with many of them. The most important thing about variant calling isn't really the caller. It's really making sure that the alignments are correct, because the variant caller is just tabulating the variant signal from the aligned BAM file. And if your alignments are bad, forget about it. For example, if you have a big insertion or deletion, BWA-MEM will mess it up because it penalizes itself so heavily for creating alignments with big gaps. Here is supposed to be a 52 base pair deletion in exon 9 of calreticulin. This is a really common mutation. It's one of the important mutations we see in myeloproliferative neoplasms, but the alignment here is awful. We do get a small number of reads with the correct 52 base pair deletion, but most of the reads show truncated weird alignments where the aligner has kind of stupidly been happier with all of these mutations and read clippings than with the actual larger deletion. So if we try variant calling from this BAM file, we're most likely to get tons of individual incorrect mutations, but we may or may not even see our 52 base pair deletion. So an important function that needs to be performed at some point to help with large indels is some sort of localized de novo read realignment using a software that takes all of the anomalous reads and compares them against each other and figures out that, hey, these are all part of the same anomaly, and then tries its best to fix them. In our lab, we use a software called Abra for this step. If we perform variant calling on this, any software, variant calling software, is now going to properly identify the 52 base pair deletion. So this type of localized realignment is really a critical step to include. Algorithms like ABRA are definitely helpful. A common algorithm that many clinical labs use is called PINDEL. It looks for regions of anomalous reads, and it performs realignment and variant calling together, but only for these type of large indels. Still, it's extremely good at doing this. The only caveat there is it gives you a separate variant output file than your regular variant calling output, so you have to deal with kind of merging them. But for certain things, it's really helpful. We use it to help with our FLT3 ITD calling in our AML patients, for example. And some of the newer variant calling systems like Google Deep Variant and Octopus, they include de novo realignment as part of their standard algorithm. So you do have multiple different options for getting this done, but it really needs to be done one way or the other. And at the end of all of this, we get a final variant list. It's usually in a VCF file, which stands for variant call format. This format has a number of unique features. We have a bunch of header lines that have information about our sample and about various information flags below. And then we have our variants listed one at a time. And for each one, we have a chromosome and a position. We have an ID, for example, a SNP ID if the variant has one. The reference base and the alt, which is whatever the mutation is, an overall quality score and pass filter if the variant caller produces that. And then lastly, a bunch of fields that show associated data like the depth and the variant frequency and the overall genotype for the sample. And a lot of these fields actually are variant caller and pipeline dependent. So VCFs from different labs may actually look a bit different from each other, but the overall format is usually the same. Now, just because we've done all these steps doesn't mean all these variants are actually reliable and correct. There are a lot of genomic regions that can really mess with your alignments and produce false positive variant calls. And it's important to recognize that and deal with it. For example, in regions where a high percentage of the bases are Gs and Cs like this, sequencers generally struggle. This is Illumina data, and you can see this waterfall type pattern of mistakes that the sequencer is making. A lot of these errors will have low quality scores, but some won't, and they can still result in false positive calls. Short tandem repeats are also an issue, whether it's single base repeats or trinucleotide repeats like this. These are sequences that are notoriously prone to polymerase copying errors. So they can accumulate errors before the sequencing or during the sequencing and give you false positive length affecting changes. And all these things on the right are local sequence-based motif errors. Another type of error can come from larger scale problems with the genome when you have multiple copies of the same sequence. That could be pseudogenes or when you have multiple copies of the same gene. Over here, you usually have solid mapping to the correct location, and the sequence just has errors. But with the repeat regions, there may not be one correct mapping location. This, for example, is a problematic exon of PMS2, which has a substantial pseudogene issue. These white boxes for our reads, they indicate a map quality of 0, where the aligner has no confidence in the mapping location. So most of this data, we don't actually know where it goes or where it comes from. And we have to be skeptical about the data that's left over. And there are tons of different ways that we can with false positive variants in this type of situation. So you need to have a handle on all these things when you design an assay and a pipeline. You don't want to be in a position of being forced to report things that you don't have much confidence in. I'm only going to say a very quick word about tumor mutational burden, even though topic that could fill a whole lecture. Just to level set, TMB is a potential marker for immunotherapy response, and it's defined as the number of non-synonymous mutations per megabase of coding territory. The reason I'm not going to go into much depth is because the pipeline for determining TMB is mostly the same as for regular variant calling. After regular variant calling, you just need to annotate the variants and remove all of the synonymous variants and then the inherited variants. And then that step, that's the hard part. If you happen to have a matched normal sample where you know all the inherited variants, then identifying and removing them is really easy. But if you're only sequencing the tumor and not the germline, this can be really difficult, and it can require some uncomfortable trade-offs. But regardless, at the end, you have a number of remaining variants, and you just divide that by the size of your assay, and that's your TMB. And as we talked about, I think in the first lecture, it's just much better if your assay is at least a half a megabase in size in order to get a robust TMB value. Okay, so that's our little pipeline from FASTQ to variant calling. And remember how I said that the alignment was really the key step? Well, it's actually more important than just for the variant calling itself, because this BAM file that we made is actually our jumping off point for doing all of our other biomarker data collection. All these other biomarker detection modules are going to critically depend on the aligned data file as a starting point for their analyses. So the alignment is really the linchpin of this entire pipeline. And now we'll go through some of these other modules in some detail and show how they work, starting with the copy number calling. Copy number calling. On the left side here is a HER2 FISH image. And you can see the green control probe for chromosome 17 and the red probe for HER2, which is also on chromosome 17. And there are clearly many extra copies of HER2 per cell here. Here by FISH, you can see that on a cell-by-cell basis. But by NGS, we're actually going to do an extraction of all of this in aggregate. Still, if you ground up this sample, you're going to see in your population of DNA that fragments from HER2 are still going to be highly overrepresented when compared with other fragments from chromosome 17 or other places. And that's going to remain true after your library prep, and that's going to remain true after your capture. And so the proportional number of sequencing reads that correspond to HER2 are also going to be elevated when you get your sequencing data back. And then when you look at the HER2 gene in IGV after you've aligned your data, that's going to manifest as increased depth of sequencing relative to other areas of the genome. So copy number calling is based on sequencing depth. Well, how is depth calculated? It's done by using the BAM file. For every region that we capture, which may be individual exons of sequenced genes, we can create a BIM. And then the calling software is going to go through every read in the BAM file. And the only thing it's going to look at here is the mapping location information. And whenever it finds a read that maps within one of these regions, it'll tally it up. There are many tools that you can use for this process. And again, I don't think there's any best tool. Many labs in the clinical oncology space use CNV-Kit, which is what we use too. It's a nice package. It allows for use with a panel of previously run normal control samples. So it's good for tumor-only analysis without having to have a matched germline sample. But multiple other softwares are good too. Like everything else, if you're putting together an assay, it may be necessary to try a few different packages to see which one works best with your data, right? Not every test is the same and not every recommendation is good for everybody. There are a lot of underlying complexities to CNV analysis because it's more than just extracting the depth data from the sample and plotting it. You have to normalize the results against the pattern that you get from normal non-malignant samples. And then there may be irregularities due to high GC content, or just because today's capture happened to work a little differently than last week's. So you may actually need to compare, today's data against both a historical baseline and against a control sample that might be built into the same run. It depends. But eventually the goal is to get the data smoothed down into a really nice looking plot. And this happens to be a really nice one. They don't always look this nice. This is a normal non-malignant profile. We can see every chromosome from 1 to 22, and each one is right on this 0 line. And this is a log scale, so this represents normal copy number, which is 2 copies. This here is a lung cancer sample with multiple chromosomal abnormalities, including CDKN2A deletion and EGFR amplification. But there are also a lot of large-scale changes like loss of the entirety of chromosome 10 here. This is the kind of stuff we struggle with in the lab because our test is built around calling individual genes like CDKN2A and EGFR. It's not a typical thing for these oncology panels to have calling thresholds and reporting criteria for all these other chromosomal alterations, most of which are of uncertain significance. But some of them really are important. Here's a co-deletion of the P arm of chromosome 1 and the Q arm of chromosome 19. This is the 1p19q co-deletion. It's the defining hallmark of oligodendroglioma. And it's such a strong association that you can actually diagnose the patient just by looking at this. So that's a project we're working on now, figuring out which of these types of anomalies is actually important to report, and how are we going to report them, and how do we set up frameworks for them. It's not totally straightforward. Here is an interesting profile. It goes up and down like a sawtooth. This is a breast cancer with biallelic BRCA1 mutations. And the sawtooth pattern here comes from large numbers of DNA breakages, rearrangements, and gains and losses that are the genomic scarring consequence of homologous recombination deficiency, or HRD. This noisy copy number pattern is a biomarker in its own right for these tumors, which many labs are now offering and reporting as a separate reportable module. You can generate an allele-specific copy number plot using a software called Sequenza, and then generate multiple different possible biomarker outputs by processing the CNV findings in different ways. This overall is a really tricky biomarker, and I wish I had more time to go into the specifics, but we only have an hour, so I kind of need to let it go. But it is a tough one, this one. So let's move on to structural variants. Structural variants involve breakages of DNA with reconnection to a distal location. The official definition is anything over 50 base pairs in size counts as a structural variant, but I kind of think that's a bit low. I generally think of these things as larger scale anomalies than that. Rearrangements, they may involve breakage and reconnection of DNA in a lot of different possible ways. So they may be interchromosomal, where two chromosomes break and are brought together, or they may be intrachromosomal. If they're intrachromosomal, they might be a deletion like this, or an inversion where a piece is switched around like this, or even a duplication like this. Regardless of the macro organization of the rearrangement, though, the biomarker itself, there's always a connection, right? And between two places that aren't supposed to be connected. And the biomarker itself is that connection point. Here we have a connection between somewhere on chromosome 3 with a distal locus on chromosome 15. And if we're on the left side, all the sequences normal, chromosome 3 until you hit this breakpoint. And same thing, if we're on the right side, everything is normal 15 until you hit this breakpoint. So the breakpoint is the biomarker. And the question is, how do you identify it? There are two main ways. First, let's imagine we have a fragment of DNA that we captured and sequenced, and it goes across this breakpoint. Under some circumstances, we're going to have read 1 and read 2 be either mostly or entirely on opposite sides of the breakpoint. So we can look at the BAM file for that. And this time we're going to look at the coordinate mapping locations of the pair of reads. So for a pair of reads from a random fragment in the genome, the mapping locations should be very close together, like I mentioned. It's highly abnormal for the left side of a pair of reads to be on one chromosome, while the sequence from the right side of the molecule maps to an entirely different chromosome. So this could get flagged for a possible rearrangement via discordant read pair mapping. That's one method. Another possibility is if the fragment is off to one side like this, so one of the reads goes directly across the breakpoint. In this case, it's a little more to the right. So the aligner gets, has put it mainly on chromosome 15, and both of them are on chromosome 15. But there's a lot of this read on the left-hand side, which is from chromosome 3, and the aligner has gotten confused, and it has issued a clipping and just basically clipped off the whole left side of it because it didn't know what to do with it. This read potentially provides supportive information about the breakpoint. So one way the software can handle this is to actually cut this read in half. This is called split read mapping, and then try independently to align both halves back to the genome. So the left half here is going to go to chromosome 3 and the right half to chromosome 15. And then because of that, we'll be able to identify, again, the breakpoint. So either individual concordant read pair mapping or split read analysis. This is a list of different structural variant collars. If you ask around at various different clinical labs, you'll get a lot of different answers about which ones they use. And many of them use multiple collars. We use here SVABA, but I would not say we're overly happy with it. And we're evaluating others now to see if we can squeeze out a little more performance. They're all difficult. Here is the output we get from SVABA for an EML4 ALK fusion in a lung adenocarcinoma. The red shows the 2 different breakpoints on chromosome 2, so that's correct. And the bracket here in blue shows all of the different quality metrics that it outputs. The quality metrics, obviously, there's a lot of them, and they can be confusing. The problems here, one of the things, this is just one out of anywhere from 300 to many thousands of structural variant output hits that this software comes up with for a single sample. It's too many to look at. What we try to use these quality metrics to filter down on only important stuff. But the quality metrics are highly variable, even if you sequence the same sample library twice. So we have ended up with a situation where we have kind of less than optimal sensitivity and specificity, which is what multiple labs are kind of dealing with. This is a particular issue for detection of fusion genes by targeted sequencing of intronic breakpoint hotspots, which is one of the things we designed the assay to do. If you're looking for things like this, which involve breakage in focal regions like ALK intron 19 over here, in this particular case, connected to intron 13 of EML4, our solution, given that the structural variant callers were not quite making it, was to write our own summarization script, which goes through all of the alignments and it picks out read pairs where one of the 2 reads maps to one of these key coordinates and the other maps to a different location. So our little homegrown software pulls out anything and everything at these focal positions that meets those criteria, which means that the sensitivity actually becomes quite high. The downside is it has a propensity for noisy false positive calls, but that's okay for 2 reasons. First, the regions that we're looking at are small, so the number of false results is generally low. And generally, it's the same easily recognizable noise repeatedly in many samples. So we actually are able to get rid of most of it up front. Second, I'd rather be aware of every possible finding. A major criteria for reporting a structural variant in our lab anyway is that the pathologist needs to be able to believe the possible rearrangement is real by actually looking at the reads in IGV. For most samples, the repetitive noise is removed up front, so the number of things that we need to review is really minimal. It can be done quickly. And by implementing this kind of strategy, using our own coding to put something together that works better for just these focal things, we've actually never seen a missed fusion in our lab in one of the key genes that we specifically target this way. So this is an example where, if you have the ability, it sometimes makes sense to code something that, isn't just designed for somebody else's research data, but it's designed for your assay. All right. Lastly, let's look at MSI calling. There are many software options out there, and most of the good ones look at the BAM file in similar ways. These are some of the most popular options. In general, when you're dealing with tumor NGS sequencing, most labs are doing a tumor-only analysis without having a matched normal run at the same time. So unless you have a matched normal, you really want an option that can do a tumor-only analysis. Some of them, like MSINGs, have a dedicated tumor-only module, and some of the other ones are more geared towards having a matched sample, but there's ways that you can fudge them to give you a tumor-only option. Not surprisingly, when we're trying to assess microsatellite instability, we should be looking at microsatellites. This is going to be just a review of how our assay does this. We wrote our own code for this before some of these other modules were available, but I think it's instructive for just showing how you might want to look at these sequences. So microsatellites, these are repeats of anywhere from 1 to 5 base pairs. And microsatellite instability means that the tumor accumulates frequent length alterations at microsatellite loci due to mismatch repair deficiency. So we need to look at these loci for length affecting changes. At least in our lab, we're focusing mostly on homopolymers, which are repeats of single base pairs, like this large poly T stretch that's adjacent to this exon of STAG2. The usual issue that you face when you do this is that these are really messy areas to sequence, like we previously talked about. So even normal samples have errors here. Here you can see a variety of mostly deletion artifact where you see reduced homopolymer length, and it's all artifact here in this normal sample. But on the right here, you can see an MSI case, and it looks a lot different. There's a lot of reads showing substantial decreases in the length of this microsatellite. So it's pretty clear that this microsatellite locus is unstable. I'm just going to show you how our algorithm works here. For each of these microsatellite loci, and this is just one locus here, the first thing we try to get a sense of the range of acceptable noise that we might see in normal samples. So we ran a bunch of normal samples and we averaged out all the signal we saw and generated one of these like profiles where 0 represents the correct length. The right side here shows how many reads we saw that are longer than normal, and the left is reads that we saw that are shorter than normal, and this is sort of the range of error that a normal sample will give us. Then we generate a similar distribution for today's patient sample. In this case, we can see a very different distribution of sizes. Then we basically mathematically overlay them by subtracting one distribution from the other. And what's left in red here when we're finished is the abnormal signal from today's sample. We just add that up, tally it up, and quantify it. Now, we then do that over many hundreds of individual different microsatellite loci. For each one, we determine an acceptable range of results that we qualify as normal. And then we ask whether the signal from that locus from today's patient sample is inside or outside that range. If it's outside, we call that locus unstable. And then we just look across all the loci and determine what percentage of the loci are unstable. And if they go more than above a particular threshold, we call the whole case MSI. This particular case on the left, this is a stable case, and all of the data from the tumor is in blue, and most of it shows up within the range of normal that we would expect from the loci that we've looked at in our normal samples, and only a few percent of them go outside the range, and that's a stable case. Whereas the case on the right has many unstable loci that are way outside the range of what we normally expect. A full 52% of these loci are outside the range, and so this is an MSI case, and the numbers that this high are what we most typically see in our MSI cases. So this approach really does a good job for us of discriminating the stable cases from the MSI. And you can see overall that this activity is somewhat similar to variant calling in that, we're looking at length affecting variants, but here we're purely looking at length affecting variants, and we're just sort of summarizing them in a different way. So now we have our pipeline, which could really be as simple as a number of commands just typed out in a little file. But before we can click go on this, we need to think a ways beyond just the software and figure out what type of computing infrastructure we're going to use. And within that, we have different choices of environments to work in. The infrastructure is just the hardware, and that could be a dedicated box sitting in the lab, or it could be a large-scale high-performance computing cluster that might be part of your company or university, or you could try to operationalize your pipeline within the cloud. The environment is more about what type of operating system or contained or managed computing environment you intend to work in. And there's really lots of options now, and they run across a wide range of infrastructure scales. And for each, we have different choices of environment that range from really easy plug and play at the bottom all the way to highly complex do-it-yourself approaches. In general, if you're using something that's off the shelf or vendor provided, like all the things at the bottom, there's very little work involved in running it, but you probably have minimal ability to customize the software or make it run the way that you want. These vendor options, regardless of where they're run, they're going to be completely locked down. On the other hand, if you go the totally do-it-yourself route, you had better have a lot of bioinformatics support, but then you can make whatever custom analysis you want and potentially have the ability to make it very finely tuned to your particular assay. When clinical NGS first started back in about 2011, there really wasn't much option besides having to buy a big server and drop it in your lab. And then you needed to install all the NGS software yourself and figure out how to run processing jobs. And then you needed to write all the pipeline software yourself. By the time I started at UChicago in 2013, we had the advantage of being able to use an existing university-managed high-performance computing cluster right in the middle here, which was great. This was a big system with a HIPAA-compliant Linux environment where all the NGS software like BWA was already installed, and it had a system for submitting jobs. The only thing we needed to do was to write the pipeline software itself, which reduced the workload tremendously. So that's us, and that's what we still do today. Although we're in the process of beginning to investigate processing in the cloud, at least as a backup. Early adopters of cloud computing for NGS analysis really needed to use it as a blank slate. They would create many virtual machines and do the difficult work of organizing those into a computing cluster. You don't really need to do that anymore. Amazon Cloud, for example, they have something they've built called Health-omics, where they have created ready-made computer clusters that you can just spin up and run your pipelines in, and you don't have to do all that work in the background anymore. And that thing operates very similar to our UChicago cluster. It's just that the machines are virtual and in a cloud system instead of in a server farm next to the university. So for us, that would be the easiest way to move to the cloud, because we could just pick up the pipelines that we're currently using and put them in Health-omics, and they would run with minimal alterations. But if you don't want to put so much effort into writing pipelines, there are many options, other options for managed cloud environments that are attractive. So there's companies like DNA Nexus and 7bridges and Illumina Connected Analytics. They all offer much more streamlined interfaces where you can log in, and even without extensive bioinformatics or coding experience, you can put together a pipeline. Even some of them offer graphical user interfaces where you can pick a software on a file and draw arrows and build a pipeline that way. Illumina in particular has an entire Dragon pipeline that will work on next-gen oncology panels, and it'll perform the bulk of the analyses quite well just out of the box. And you also have the option to modify it or add modules to it. So these are some of the newer frameworks that probably make the most sense for a lab that's just starting out, particularly if you don't have a huge amount of in-house bioinformatics support. So assuming we've picked an optimal computing structure and we've run our pipeline, the last thing to worry about is interpreting and reporting the case. To do that, we're really going to need a system that can help manage the process, right? That system should have a database where we can put in clinical information about the patient, and then we should be able to load in the variants we got from our variant calling operation, as well as all the other biomarkers we extracted during this whole pipeline operation. And ideally, under the hood, the system should have access to mutation and therapy associations, so that if we find an actionable mutation in a tumor, we can provide a treatment recommendation. And also information about clinical trials would be nice in order to hopefully make recommendations about those. Not every lab does all of this, and some aspects of this we're still working on as well, but these would be what you would try to do. There are a number of options for systems like this. We started a long time ago, so we ended up making our own, but there are off-the-shelf options from places like Purion DX and Genome Oncology and Illumina that labs can use without having to do like a dreadful development project like we did. But there are costs associated with using a commercially available system, and it may not be perfectly customizable for your lab and your assay. The system should have a view for reporting and interpreting variants. And here you can see that the variants we've loaded in here are annotated, meaning they have additional metadata about them. In our lab, we annotate our variants up front before we upload them to the system. But if you're using Purion or Genome Oncology, the annotation is performed within the system, and you just drop in your raw variant list. A variety of different databases are needed in order to do the annotation. Gene and transcript information are needed to assign genes and exons and coding changes to mutations. And you need to follow the published rules of the Human Genome Variation Society in order to have your annotations in the correct understandable format. You need databases of inherited variants like Genomad in order to be able to flag variants as common inherited polymorphisms. And lastly, we should be either be able to bring in or have easy link outs to resources like Cosmic and OncoKB and ClinVar. Cosmic and OncoKB, they're really helpful for giving information about common somatic mutation findings. And ClinVar, even though it's a database of inherited findings, is still extremely useful because if a variant has been seen before as a germline event, it helps us to understand its function. So given all this information, we can try to go through the process of interpretation. There are some real differences in opinion about how to interpret and report somatic genomic findings in tumors. The major published frameworks are the AMP, ASCO, and CAP criteria in the US, and the ESMO Scale of Clinical Actionability for Molecular Targets, or SCAT, in Europe. Both frameworks are similar in that they are primarily based on clinical actionability. For example, the AMP framework involves grading variants on a scale of 1 to 4, with 1 being variants with the highest level of clinical actionability either FDA-approved therapies for patients with the right tumor type, mutation combination, or else strong clinical trial evidence. Level 2 might be if there's an FDA-approved therapy, but it's for the same mutation in a different tumor type. Tier 3 is for presumptively somatic mutations that are not known to be associated with cancer. And Tier 4 is for variants we think are benign. So these are your variants of uncertain significance. Over here, the SCAT system is similar. We have tiers 1 through 5 for decreasing levels of clinical evidence, with 5 being for variants with minimal drug-related information, and tier X is for variants where drugs have been tried and failed. And I'll be honest, like I have some problems with both of these systems, and so do many other lab directors. First, making tiering decisions solely on clinical actionability can require some difficult judgment calls. So even if everyone in your group is 100% up to date on all clinical trial results, there is still a risk of interpathologist disagreement, and then you don't have a great standardization of your reports. Also, a clinical picture changes rapidly. And so a report that I issue today could be seriously out of date in only a few months. And perhaps the most frustrating thing is that there's really no proper place to put many truly important variants. Let's just take a little example of a MIB-NFIB fusion that's found in an adenoid cystic carcinoma. This is a salivary gland tumor. This particular fusion is a disease-defining event found in nearly all cases of this tumor. So it definitely doesn't fit into tier 4. There's also plenty of evidence of cancer association, so it can't be a tier 3 either. But there's also no experimental therapies or trials for this fusion, so it's not in tier 1 or tier 2 either. So here we're in a position where there's no ideal place for an anomaly that is the main molecular driver of the tumor and also an important diagnostic finding. In our lab and in many others, we instead base our variant classifications around pure biological pathogenicity. So if it's a main classical tumor driver like MIB-NFIB or a KRAS mutation or a P53 mutation, it's tier 1, regardless of the latest therapy information. If it's less clear if it drives the tumor, but it's still a likely possibility, then we grade it as likely pathogenic or tier 2. And if it's probably somatic, but we can't find any pathogenicity information about it, then we consider it tier 3 or variant of uncertain significance. Then, when we write our report, we layer in other information like the diagnostic ramifications and the prognostic information and clinical trial and treatment information. And we find it much easier to grade variants this way. And we get, I think, higher levels of agreement between all the faculty in our group. Also, a classical KRAS mutation is going to be a tier 1 today, and it'll be a tier 1 forever. So our reports are, less likely to go out of date. And perhaps most importantly, I feel like this kind of system is a bit more in keeping with our role as pathologists, because a large part of what these panels are about is real pathology practice, helping to make the right diagnosis for the patient. And so our reports should focus both on treatment possibilities and also on what's important from a pathology standpoint. Okay, well, that is the end for me. Thanks so much for joining me for these 3 lectures, where we went all the way through the process of these NGS panels from start to finish. It was really a fun process for me to put this together, so I hope you enjoyed it as well. And I'm happy to take any questions. Thank you.
Hi, everyone. On behalf of LabRoots, I want to apologize for the technical difficulties that we were experiencing. The on-demand version will be fixed. But with that, I want to thank everyone once again for joining in on this webinar series. It's been a pleasure to moderate this. And also thank you once again, Dr. Segal, for your time and effort and your invaluable insights. So I'm going to open it up to the audience for any questions they may have. And the first one that we have is, when trimming, I find that a large portion of good reads are lost. How do you substitute those poor quality bases?
Yeah, I saw that one. Before I start, I just want to apologize to everybody. I'm sorry. I hope you didn't go nuts listening to me repeat stuff. That was not the plan. In terms of trimming, I don't know exactly how to answer this question. When we're doing trimming, we're basically just trimming adapter sequences. And so we're trimming using a software called Trim-O-Matic, and we just give it adapter sequences to trim off. And I don't believe that it does more than just trim the adapter sequences. And I don't think it trims based on the quality. So I'm not sure what software you're using, but it might be that you need to try some different trimming software if you're losing like good actual components of the reads. If you're just, maybe you're just losing the adapter sequences, and that's okay. But I'm not sure about that. I think maybe trying some different trimming approaches could work in different software.
All right. Another question, how can bioinformatic analysis or NGS data be integrated into existing clinical workflows? For example, do you integrate other data into your NGS report, such as, maybe PCR data, IHC data?
We don't, but other people do. I think the commercial labs tend to do that a lot more than the academic labs. Like in our group, our IHC group is entirely separate, and it's part of surgical pathology, and they do all the IHC and they put that into their surgical reports for, as addendums. And same with fish, that's a different group. And so it's kind of the level at which it gets integrated, I think, is a little bit different often in the academic labs versus what happens at one of the larger commercial labs where they do all that together and throw it all into the same report. So it can be a little bit more challenging on the academic side to get everything integrated. Then again, you're not duplicating stuff. At some of the commercial labs, they're going to redo IHCs that you've already done in-house and then build a patient for it again. And I don't think that's so great. So both models have some issues.
And then how do you minimize the complexity of the data in a report? And does your lab do any kind of internal education on how to read a report?
Getting the right amount of data into the report is difficult. We are in the process right now of trying to figure out how, what our next version of our reporting is. Like in our old lab information system, we were really stuck just being able to use only like text-based reports. So that's all we got, text-based reports. And we're working now on a PDF format because we're using Beaker and we can actually make PDFs and put them into the system. And so it's like, what should that PDF have, right? And how do you make it so everything is sort of easily viewable? And how do you bring in other features into it, and without making it too complicated? And it's not easy to do. I think I've seen all sorts of reports from different labs, and there's a huge variety there. And making it visually appealing and simplistic enough that it can be easily read is really a challenge. I think delineating separate clear areas for the mutations, as well as maybe treatment recommendations, an area for the pathologist to describe their what they're seeing, and give some sort of personalized report. I think is important. Visual graphics are helpful as long as they're not too cluttering. It's really difficult. For us, in terms of education, it depends on the test. Like for our large comprehensive panel, we pretty much are I think everyone's kind of used to it on our report format. So we don't really have a whole lot of times where we need to educate people on how to read them. But, for example, we just did a brand new, NGS panel for minimal residual disease in our heme malignancy patients. And that one is a challenge to understand what the meaning of all the results are, because you might have a mutation and in one patient it's significant and in another patient it's not. Based on whether that's a mutation that they had, before treatment, et cetera. So how to display that, we're still working on, because we're putting together a PDF report for that one too. But in the meantime, we have like a working group meeting where we meet with all our heme oncologists on a bi-weekly basis, and we go over reports, and we just make sure we're kind of on the same page about what they mean and how to follow them up. So it's really sort of a longer term educational process for that test. So I think it kind of depends on the test and familiarity of the oncologist with your report formats and your reports, and you have to sort of play it by ear a little bit.
Do you also have like a patient report, like a report for patients that they could view, or not at this point?
At this point, no, it's just the same report, and I think that's kind of standard across the board.
Yeah.
That would be a difficult report to write, because you want, again, then you'll really want to, I guess, simplify it, but you still want it to retain all the information that it has to have. I'd be scared, honestly, to try to put a report like that together, and try to thread that needle between putting in the things that need to be in there, in the complexity that they need, along with what the patient should take away from it. I kind of feel like I'd almost rather just issue just my report, which we try to write it so it's somewhat approachable, but then that's when they should talk to their oncologist, because other
Right.
than that, like, I think there's a lot of room for misinterpretation and the patient to take the wrong message home from it. So that'd be a scary report to write.
Um, Kind of in this vein, another question, are there workflows available that need little to no bioinformatic or variant interpretation experience?
Um, mm, yes, I guess. Interpretation experience, in terms of, you could buy an off-the-shelf product, you could run it. There are, uh, you know, off-the-shelf packages for variant review and reporting that you can use. I believe some of the companies, if you work with them, will provide their basic language for every variant. You might find that some of it needs tweaking or adjusting. I think it would be challenging to report with zero experience. I think you could have services that will significantly help to reduce the variant review burden, but I still think you want somebody with experience who's looking it over before it goes out. Otherwise, um, I, you're sort of completely leaving that up to a third party. And maybe if you find a third party that you really trust, or like maybe there are third parties that do like professional level interpretation and really stand behind it, and but that would be tricky for me. I think somebody needs to, at the end of the day, like be able to say this report makes sense. And if your assay is small and targeted towards the hotspot things that you're just reporting the same things every time, then you could generate it. If it's just KRAS, BRAF, and a few other things, you could have a lot of your verbiage ready to go within like a short amount of time. And then you could be running stuff, and then it wouldn't be too challenging to report. The more genes that you add, the more you tend to have to start to look things up, and there's more like, this is a weird tumor with a mutation that we haven't seen before, what do we say? But if you really focus on the bread and butter, I think you might be able to get to a point where the day-to-day variant burden isn't too challenging. But yeah, that would be my advice on that.
Great. And then one is on variants of unknown certainty, I think it is. And how do you search for the actionability? And I guess how do you report those?
Yeah, it's tough. I mean, we see this all the time. You do a case, you get a variant, and you haven't seen it before. That phenomenon is true at a multi-hundred gene panel. Is true in every single case, um, and it's a process of looking it up in various different sources, right? Like you want to see if it's a common mutation. You can look in um, Cosmic or CBioportal. You can try to look in OncoKB, although that's really more for the things that have been seen more frequently. Um, ClinVar is often helpful because there's a lot of these mutations that have been seen in the germline setting, and then you happen to see it, and then if there's a patient who's affected, then that variant is actually like doing something. And other than that, you really have to use a combination of search features, even, PubMed, Google, other types of things to try to find literature about that particular variant. And it can be a real challenge. So that's like really, it's those new mutations where, things take a lot of time. So like I've been involved in a multi-institutional consortium of academic labs, and one of our big projects is putting all of our lab databases together so that hopefully we'll be in a position where if we have a new variant, we can ask, did somebody see it at another institution? And at least, maybe we can reduce that kind of volume of work. If, a different institution's seen it and they've found some literature on it and they've reported it, then we can, take a little less time looking at it, and we can use, their previous work, because that really is the largest bit of work that goes into variant interpretation.
And some more questions here. One question is, how does PCR duplicates impact copy number analysis?
Yeah, that one is tricky. I don't have the perfect answer for that. I think we've tried copy number calling with and without it. My impression is at low depth, the PCR duplicates aren't, you'll have some number of PCR duplicates. And as you sequence more and more and more and more, eventually you exhaust all your different start and stop positions, and then you start throwing away more and more and more reads. So I think that sort of attrition happens more when there's a copy number gain. Instead of a really high depth, it brings it down a bit. And I think we actually use PCR duplicate remove data for our copy number calling. I don't think it has affected us too much, because the blunting, I think, happens when the depth gets really high. And so you're still going to get like a very high number, even using PCR duplicate remove data. But there's a good argument to be made for using non-PCR duplicate remove data. I think you can do it either way. It's possible that non-removed data is better, at least, but what you're going to get is, instead of having a cap on the level of amplification, which we probably have, you probably have a more dynamic range of high. I don't think the lows will be any different, but the highs will be different. But from a clinical reporting standpoint, I don't think it matters all that much. But I don't know the exact impact of it on low-level gains. I would assume it's kind of minimal.
And one last question before we close out the Q&A. What measures should be taken to ensure patient data privacy and security during the bioinformatic analysis process?
Yeah, you ideally want to have, you kind of should have a HIPAA compliant service. And for us at University of Chicago, we use a high performance computing environment that's HIPAA certified. So the team that manages that resource has gone through all the documentation and stuff in the background to be able to say that this service is HIPAA compliant. And our lab information system runs on virtual machines in that same system. But it has been a back and forth, because those are on the academic biological sciences division side of our university, and they're not housed in the hospital. And it has been, an ongoing discussion of whether all of our stuff should be moved onto proper hospital systems, which would be fine, except, right, we use a high performance computing environment that has all the next gen sequencing software already there, and it's really made for running large scale pipelines, and the hospital doesn't really have anything like that. So it's not super easy to think about how to move that into a hospital environment. Our lab information system, yes, that could be moved into a hospital environment. It hasn't happened yet, but it's still on a university level HIPAA grade system. So you definitely want that kind of protection. You don't want to just run it anywhere. I think, even if you have a server that you've built in the lab, that's probably a concern. We had our NovaSeq, or actually back in the old day when we had a HiSeq, we had our HiSeq hacked by Russian hackers. But fortunately that was, limited in scope. They just, they just encrypted everything on the machine, but we had a backup. But at the time, there wasn't a lot of protection for the sequencers themselves. But you got to, I guess, worry about the lab. So it's probably best to have it in an environment that's protected. If you're going to work in the cloud, you need a business associates agreement. That's typically how it's done, and then the company assumes some liability in the event of a HIPAA breach, and they do their best on their end to make it secure. I think largely that's a very secure way to do things. I don't know exactly what they do on their end to make it HIPAA compliant, but they're at least certifying that they're doing that and taking on some of the risks. So, if we were going to use like health omics through Amazon, we would get an agreement like that signed, so we would be able to do it. And we're kind of working through some of those types of agreements now, but you need that in place, and I wouldn't do it without that.
Are more clinical labs moving to cloud computing?
I think so. I I know a few that have. Yeah, I think a decent number have, and I think it's going to accelerate. It was, when I started working at U Chicago, it was kind of difficult to use the cloud for this type of stuff. But now there are these like built, functional environments, and you can contract with a company that has an environment that you can work in, and it's much easier to like make a cluster of machines that can operate. We used to kind of have to do that manually, I think, and it was really challenging. So like 12 years ago, like that was a really challenging job. Now today, they help you do it, and they, make it pretty easy to spin up something like that. It's still a challenge to move to the cloud. Like we're working on using that at least as a backup. And so our bioinformaticians are plugging away at that, but it's still a learning process. It's still a different way of doing things, and it still takes time to get everything set up. And so it's for us, I mean, we're not devoting like full-time effort to it, but it's a multiple months long process of kind of getting things set up so that we can run stuff. And then you want to be able to run it at scale in the cloud. So it takes effort to get it set up. But I think once you get it set up, it's probably relatively straightforward. You don't have to worry about your systems having downtime or whatever. You've got sort of unlimited bandwidth. You can run as many samples as you want. So there's some real advantages to it. And our high performance computing system at our university, every few years it gets traded out and they build a whole new one. And then you have to deal with, how does this new one work? And how do we modify our pipeline so it works with this whole new system? And in the cloud, you won't have to do that. So, as it gets simpler and simpler in the cloud, I think more and more people sort of migrate to it.
Well, thank you so much. I want to extend our sincere gratitude to you, as well as to LabRoots and Roche for making this event possible. So we're going to wrap up the Q&A portion here. We want to extend again our gratitude to the audience for joining along with us during this webinar series. And if you happen to have missed any one of the webinars, all will be available to you on demand through LabRoots. And right now you can find them within the auditorium. You can click on webinars 1 and 2 individually, and we'll have the third edited version available soon. So thank you so much.